Okay, well, I think we can start now. Thank you. Good afternoon, everybody. Thank you for joining us. Always faithful these sessions of for journal clubs and discussion of nice and interesting papers. Today I am your. I was supposed to be the spare host, but now I am the first host. And I hope that Ruben has not any issue, any problem with the connection or else, otherwise. So the topic of today's discussion was chosen by Masimba. I think I was not privy to the selection, but I imagine. And it involves genetic architecture and spatial electrical biomarkers for cardiac arrhythmias. I must say that I gave a look at the paper a couple of times, and I really need more detailed presentation. It spans quite a lot of things. So, well, without further ado, I leave the floor to Masimba for his presentation. Thank you.
Oh, okay. Thank you, Guido. Okay. So I don't know if I will be able to add a lot more detail to the paper. I thought you would be more comfortable with this because the paper is mostly about genetics with something that has to do with electrical biomarkers. So I will try my best to present what I understood and point out a few things that I found very interesting.
Take it away.
So
the only thing is that, for example, I don't know if everybody sees your screen, but I see just a black box.
Really?
Yes.
Yeah, I do not see your presentation.
Okay.
Oh, okay.
Much better.
So,
yes.
Oh, now you can see it.
Yes. I don't know if it's presenter mode or when you go in full screen. Now I can see your slide.
Okay, so is everyone okay with this setup? Because when I go into full screen, then it seems that it goes black.
That's okay for me.
Okay, so I suppose a lot of you are familiar with this idea of a cardiac vector graph. So this is basically what they say that the cardiac vector graph, especially the spatial one, it's a three-dimensional representation of activation and repolarization. And especially in the three-dimensional one, the one entitled spatial QRST angle over there, the angle between the plane of the repolarization in blue and the plane of the activation in orange seems to be a kind of marker for cardiac diseases. And also, there is a two-dimensional version of it, which they call the frontal QRST angle, where it's just the angle that you see there between 60 and 30, which is also the angle between the activation and the repolarization. So what you will see in the presentation, it will become very apparent, is that it might be better to use the spatial QRST angle than the frontal QRST angle, because the spatial one contains a little bit more information that is relevant to what's going to be discussed in the paper than the frontal one. In fact, the information in the frontal one is wholly contained in the spatial QRST angle. So they say also that, so this is what I just said. They say that there are these markers, these angles, and it's not well understood the biology that drives these the abnormal observations of these markers. What's very interesting is that it has been shown by some studies that the spatial QRST angle, if it's abnormal, it suggests a 5-fold risk of cardiac disease or cardiac death. And in the paper, they mentioned that there is no other marker on the planet that's better than this. I don't know if that's true. But if it is really true, that's very interesting. And it might actually point out to something that I'm kind of a zealot about, that it's better to use three-dimensional information, to take the three-dimensionality of the data that we get from ECG or high-density body surface mapping to make diagnosis or make some studies than just to take the individual sensors and then look at them in isolation. So anyway, going back to this, they wanted to obviously understand the underlying biology, and they did this with what they call a genome-wide association study. So the idea behind genome-wide association studies is that they can be used, especially in instances where there's no predefined hypothesis of what is going on. So you do this analysis, and then you get back some information which will probably point you in the direction of what is going on. Yeah, so this there has been a genome-wide association study that was done with this many individuals that identified 3 independent gene loci that were associated with cardiac development and cardiac conduction. But this study was small, so these guys, they thought they could do better, and they took this many number of people, which is about 10 times more, to do the genome-wide association study. But it was a meta-analysis for the spatial QRST angle, and then had about 150 individuals for the frontal QRST angle. And they say that this is the first genome-wide association study for the frontal QRST angle. So what did they find? When they did their analysis, they found that there are 61 lead variation, genome-wide significant lead variants of genes that reproduce an abnormal QRST angle. They found 61 of them, of which 58 were new, and the 3 were those ones that had been found from that smaller study. And they found that for the frontal QRST angle, there were 11 gene variants that resulted in an abnormal QRST angle. And of these 11, they are all a subset of the 61. So this is the point that maybe it's better to use the spatial QRST angle, because the frontal one is not going to give you any more information than the 3-dimensional one. And so as you will see, I only presented the results for the spatial one, and that's what they did also almost exactly in their paper. They have a very small section that discussed the 2-dimensional angle, because they said, okay, maybe when you go to 2 dimensions, you lose some information that's in the other planes. So what did they find? They found that they did this from their results. They did this Manhattan plot, and so these genes that are annotated NFIA, HAND1 and TBX3, these are the genes that had been the gene variants that had been identified in the prior study that identified 3 ones, and these are the almost chromosomal locations of those genes. We will get a bit deeper into what this means, and this is the one that they obtained for the frontal QRST angle. So these are the genes, gene variants, and then the chromosomes. And you can see that this one, for the spatial one, there is really significantly more data than in the frontal one. So they found out that from these 61 loci, 11 of them, they really were expressed in the traits of the left ventricle, the right atrial appendage, and the some vascular arteries. And so this actually points to maybe that some of these genes, they may have something to do with the development or the continued existence of these parts of the heart. So to predict these effects, the effects of these 11 genes, their expression, they did this, what they called this transcriptome-wide association study. And what they found was that 33 genes in general were significantly associated with the with the spatial QRSTA angle, with 26 of them being within the loci of the 61 that were observed. And of those 26, 10 were impacting multiple cardiac tissues. So of these 33 genes, they identified that, so the genes on the right, if you know them, sorry, on the, is it the right or the left? I don't know if you can see my cursor. They observed that some of the genes, when they were expressed more, they would cause an increase in the spatial QRST angle, whereas the other ones, they would reduce the angle the more they were expressed, except for one, this gene, which they identified that if its expression was increased in the aorta, this would coincide with an increased spatial QRSTA angle. Whereas if it was expressed more in the ventricles, this angle would be smaller. So what the so we said that 26 of the genes, they are coding genes, as they say. So then for the other genes, they supposed that they play a regulatory function within the heart. So maybe some processes are going to be impacted much more by these non-coding genes, and depending on how they are being expressed, they will affect maybe ion channels and stuff like that. So they identified that 17 of the loci had promoter interactions in the left ventricle and right ventricle tissues. So something that is quite interesting that they observed, that they try to do, they try to associate these angles with non-ECG markers, which are used clinically, because these angles are not yet used in clinical diagnosis. So they tried to associate the abnormalities in the angles with cardiac conduction. So they looked at the PR interval. Then they looked at the ventricular depolarization through the QRST duration, then the repolarization with those intervals. So what they found was somewhat very interesting. So they have this correlation matrix. The upper half of the matrix is the phenotypic correlation. And then the bottom half, the lower half of the triangle, is the genetic correlation. It's quite surprising that the phenotypic correlations are almost non-existent. between these angles and the non-ECG markers. I find this really interesting because I would have thought that there would be a strong correlation between the phenotype of the angle and the ECG markers. Also, the genetic correlation is poor. They actually just remark that there might be some kind of genetic correlation between the PR interval and the angles, because it's the one that seems a little bit more significant. So again, this brings to question, like, do we get more? Should we still be looking at these ECG markers as we do right now to make them be the theme of ECG, or we might need to look at other things? Because remember, the spatial QRST angle, if it's abnormal, you have a 5-fold risk of developing a cardiac disease or dying of a cardiac disease compared to someone who has a normal QRSTA angle. So anyway, I found this interesting. So in terms of, so they then looked at the genes from other meta-analysis and genome-wide association studies that tried to link gene expression or gene variants with the phenotypes of the ECG. And this is what they found. So these are all the 61 variants, lead gene variants that were obtained from the study that they did. So you can see that a lot of them, they are also involved in some other markers of ECG, even though the correlation is weak, as we saw from the previous slide. And there are these, I think, 7 or 8 gene variants that are in the little gray square on the corner there. These were only associated with the spatial QRST angle. And if you really think about it, when we look at these intervals, like these other ECG markers, they are only containing a subset of these genes, whereas the QRSTA angle is associated with all of them. So again, I find this interesting. Could it be just that looking at the data in a 3-dimensional way is giving us more information to dig through? Or maybe we have been looking at these intervals in a slightly not so nice manner. Okay, something to think about. So what they managed to do within the study was that each gene loci that they had, they managed to assign an effect size to each. So in theory, just by someone's genetic profile, you can compute what they called a genetically determined SQRSTA angle. And when they did this computation, they wanted to see, do people who exhibit some diseases, can this genetically determined SQRT angle point us to certain diseases? So what they found was that it was it increased the odds in abnormal genetically determined SQRT angle was associated with an increased odds of vascular or bundle branch block, but this was already known. This was reported a few years ago according to their paper. And unfortunately, they found no causal relationship between an abnormal SPQR-ST angle and some cardiomyopathies. So up to now, there was this momentum that, okay, maybe there's something here. But unfortunately, they could not make this correlation. So they then really looked into the function of the genes. They identified that a lot of them were involved in cardiac development, muscle cell differentiation, cardiac contraction, and actin filament-based movement. I don't know what this last one is. And now, okay, so this is what I just said, that 7 of the loci are unshared with ECG based loci. And what they observed was that only a few of the genes that they identified through these angles were involved with cardiac ion functions compared to ECG traits. So we will, I think maybe it's, there will be a discussion about this a little bit later. So they also noted that maybe the widening of this angle might just, okay, sorry. Yeah, so this is it. It might just reflect some intermittent ventricular changes in conduction, but these might be not changes that are obligatory or conditional to these cardiac myopathies. And considering that most of the people here are interested in sudden cardiac death, this might be quite interesting, that maybe what's causing sudden cardiac death, in most cases, these are just intermittent problems. They are not there to stay for long. You cannot do most tests to figure out what's going on. So maybe this QRST angle is something of interest. So they say that previous studies have shown that this angle, the spatial QRSTA angle, reflects abnormalities in ventricular repolarization that are a result of abnormal depolarization. So this could also explain why there are no, why you don't, there's not really a correlation with these ECG markers, because the ECG markers, they are more about conduction or channel function. But once the depolarization is not working very well, repolarization is not going to work so well. So this is like a downstream effect of the abnormal depolarization, rather than the case where the depolarization is normal, but it's the conduction of the heart that is a bit problematic, and things show up in the ECG a little bit better. So they also checked the heritability of these genes. They observed, so again, the value of the two-dimensional thing is not so much, because even the heritability of the genes is lower. You first have fewer loci, and the loci that you have, they are less heritable than the ones that you get from SPQR-STA. So again, should we concentrate on two-dimensional stuff, or maybe let's move to higher dimensions to take full advantage that we have this information? Let's just use it. Okay, so this is the point that they made, that you lose some information when you just project your things to a two-dimensional plane versus dealing with things in 3 dimensions. So this is something that they discussed in the paper that I couldn't find a very good way to present without making a very bad slide. It's that most of the non-coding genes that they observed, they have a disproportionately high impact in the development of fetal heart tissue. Okay, basically in the development of the fetus, because there is a graphic that they have that actually that shows or strongly suggests that it's the fetal development that's really affected compared to things that happen in your adulthood. So it's likely that people who have these abnormal angles, this is something that goes wrong when they are still fetuses, not that goes wrong throughout their life. So what they say is that this once something goes wrong in the fetus, this could predispose you to having poor development of your cardiac tissues or poor recovery once you maybe damage your tissues. So let's say you have a heart attack. Your recovery is going to be poor when your SQR ST angle is abnormal versus to someone who is a one that falls within the normal range. So this concludes the talk that I had. Thank you.
Thank you, Masimba. Quite a lot of information. Personally, I'm not sure. I'm sure I have reviewed the methods. I'm sure that with these large numbers and this kind of studies by proxy on leads and lead variants and transcriptomic phenotypic wide association, I mean, everything is properly conducted. On the other hand, like when I hear that there is problems at the fetus level, to me, it's reminiscent of the old theory of dedifferentiation during cardiac insults. So this might be also an opening that is not per se influenced by hereditary of the variants. I'm not sure if we can take into account like acquired variants or inherited variants. It might have been useful for us to have an overlook of the studies that they are pulling together to assess these conclusions. As far as I understood, this kind of angles of solid angles can be determined by 12 lead ECG already.
Yeah. So they take a 12 lead ECG, then they kind of plot it in a 3-dimensional space.
Taking into account, of course, that we're looking at the ECG macroscopically, and we're not considering a whole lot of things that goes on in the heart, of course, because the ECG contains a lot of information, but it's only one slice of the cake. How do you suppose? What's your opinion on why there is so little phenotypic correlation with these angles? It's a curiosity for me.
I don't have a very good explanation for that. I was actually surprised when I saw it, because really they are saying this is representing repolarization and depolarization, but it could be that maybe the way that it's done for ECG, it's to really look at like you said, a slice of the cake. You're not looking at the whole cake. So maybe by looking, by accepting, in a sense, I saw the formula of how the vectogram is computed, but I have not delved into like why they chose those parameters. So the vectogram, the cardiac vectogram, for me, it's really taking the spatial relationship of the sensors into account versus just looking at single traces and saying, oh, look, it's wider, it's thinner, it's shorter. Yeah.
But for me, it's like, could you confirm to me like that in general, a smaller or non-uniform angle or more uniform angle is predictive of cardiac health? I imagine it's like I make an association with.
Yeah, it's difficult to say because I think people have been only interested in the abnormal one. Okay, I don't even know how they came up to decide what's abnormal and what's normal.
I see.
Yeah,
I mean,
the vector cardiogram is supposed to give the orientation of the heart dipole over time.
Okay.
Right.
Okay.
Is it better if this solid angle is smaller or bigger?
Who knows?
Okay.
Yeah.
There's nothing you can say a priori.
Okay.
Yeah.
And there are
but if you
data on healthy volunteers, against this screening program, so they don't calculate this. I've seen that it's basically something that was very popular 10-15 years ago, and now it's resurging.
Yeah.
All right. That's
yeah.
interesting.
The concept is.
You can say that a large QRST angle is good because the T is supposed to be a small QRST angle is good because the T wave is supposed to be concordant, and if it's small, then usually there's something wrong.
Okay, so there is a range. There is a range of health. So let's say so.
Yeah, I say it very, very coarsely. It means that the inverse relationship between action potential duration and activation time is lost.
Right.
And somehow that is not good.
Somehow, but we don't know.
We don't know how.
We don't know why, because there's no reason. There's no constant action potential duration over the heart, so there's no necessarily a pattern.
No, but that somehow it seems that somehow the heart learns what the activation order is, and it adapts the APD so that the later activated cells have the shorter APDs, and that may be protective against arrhythmia.
But not if you look at, well, it depends what gradient you look at. Not the apical basal.
No.
Transmurally, yes. Apically basal, no.
No.
So it's very complicated. I don't think we still
say maybe. Okay, sorry. I just wanted to say maybe things like these branch bundle blocks are easier to observe in the angle. Like Ed was saying, that it shows you the orientation. So if one side of the heart is not depolarizing at the same time as the other half, this dipole is going to point favorably in one direction more than the other.
Yeah.
So maybe for
these, of course, if you have an acute change in depolarization order, then the repolarization order cannot adapt so quickly. So then you can get anything.
Yeah.
What I want to say, like, if you have a left bundle branch block or right bundle branch block, you can see it from the 12 lead ECG. You don't need this. You don't need this reconstruction.
Yeah.
I'm very said about this that it can potentially be a print of something that is transient. So maybe if somebody that works on sudden cardiac death has a more pertinent question on this, please go ahead because, as you know, I work on mostly on superventricular arrhythmias.
Yeah, well, we don't know anything, so we are just throwing spaghetti at the wall and seeing what sticks. So
Yeah,
there's no harm
if
in
people have
trying.
a bundle branch block, then they have such a long activation duration that the APD learning mechanism cannot adapt anymore, and then you cannot expect them to have a small a small QRST angle anymore. So even if they have just the left bundle branch block and their heart is completely healthy otherwise, then they still have a different angle.
Okay. Yeah, so maybe the other thing is the issue of sensitivity, that maybe with these angles, you get a bit more sensitivity than with the traditional ECG. So it might be better.
Okay, but I understand that this is there. I've seen that there is some data on hypertrophic cardiomyopathy, but yeah, I guess it doesn't make a lot of sense to look at it on ischemic cardiomyopathy because I mean here there's massive structural damage. If somebody wants to intervene, please go. I'm looking at another figure, and I want to ask a question on figure 6, which I don't think it was in the presentation. So, but please go ahead.
Yeah, if I may have a kind of curious question. So when like looking like just or like quickly skimming through the manuscript, I noticed there are, I don't know, like 60 authors or how many. So I'm wondering, because those people are, I guess, contributors to the data, that many centers contributed to the data and then they pull the data. That's how I imagine it was. So is there any characterization, like who is the population we are looking at? Like there are like 100,000 people, 150,000 people. Like where do they come from? Like what is the ethnic background? Like these kind of things. Is it somewhere in the paper described?
Okay. Yes, it's written in the paper. It's written in the paper. This is not how can I?
We're losing your microphone.
They don't talk about it much. They don't talk about ancestry specific studies more. They really focus on the study that 81% of people of European descent. They I think 10% is people, I don't know what that means, and 7% African. So they did so you can see that it's heavily weighted towards people of European descent.
I didn't get much of what was said. I understood that there was a prevalence of European descent. Is that right?
Okay.
Okay, but I have.
Okay, should I repeat?
Can you show figure 6
yes,
on your computer?
We had this. Yeah, and if you can show us figure 6 as well from the paper, please, so we can look at this phenotypic association of alleles.
Okay, how do you do that? Do I stop sharing?
Yeah, you can stop sharing.
I need to add sharing. No share. Okay. I'm like an 80-year-old person. I don't know how to use computers. Okay, days 6.
Yeah.
You can see it.
Yes. So you can see This is like
I don't know what it means.
this is phenome-wide association study, and it's like it goes backwards to associate particular alleles with diagnosed condition or known conditions, but I don't really understand these alleles. They come from where? And because there is one which is sufficiently associated with protection from cardiac arrhythmia, because I see that the odds ratio are below 1. So this RS2723294, where do they get this? There are like loci that appear.
The things that maybe I should have led with this. I have a net background. I just found this.
I'm sorry, Massimo. We cannot understand. We cannot understand anything with your mic.
You can't understand anything.
No, it's all broken.
Can you hear me?
Try to talk. It sounds pretty much the same.
Okay, I will try to log off and then go again.
Okay. All right. Let's see how it goes. I think I want to share my screen just to have a look at this.
Okay, I am back. Can you hear me?
Yes, much better.
Okay, much better. Okay. I was saying that maybe I should have confessed my sins before. I have a maths background. I just found this very interesting. So most of the things I had to really learn from the physics.
No, I'm sorry. We have the same.
From my understanding,
We have the same issue back again.
Okay. I don't know what's happening with
Now
this
it's
computer.
clear. Now it's clear like sky, like the sky in summer. I don't know what you did, but now it's clear.
Okay. Okay. I don't know what I did as well. So I was saying that I should have maybe prefaced the. Sorry, it's still okay.
Yeah. Yes.
Okay.
Please.
I should have prefaced the presentation by saying that I have a maths background, not a biology background. I just found this paper very interesting. But to my understanding, they have this database, UKB. I think so from the UK, where they have their genetic, basically their entire medical histories, so they can make these correlations, basically. And some of these genetic things, it's well understood what the genes themselves do. So I think they are coming from that angle and then saying, oh, look what we found here.
Right.
Yeah.
Is this I'm just fascinated by this because it seems like it is at least in figure 6 there is only one lead variant. No, sorry.
Okay.
7, yes, on 7 on this position. Well, it seems like there is some lead variant that is associated with a phenotype, and that they are dependent from this spatial QRST angle, but they seem to be cardioprotective.
Yeah.
More than a risk factor. So this is fairly interesting, and it seems to be.
So yeah. So in the include they actually mean that some of the that most of the genes that they observed from Alice, they have a lot of they impact quite a number of tissues. So they like there is this part where maybe it's not here. But anyway, what I think I would do is I will show you the supplementary data, which is quite interesting in itself. There is a figure that I found very interesting in the supplementary. I'm just trying to get.
I'm so sorry. I'm doing my best, but I really cannot understand a word of what you're saying. I think there is really a problem with your microphone.
Okay.
But anyway,
yeah, it's more maybe from the
no. Okay. Well, I think that this paper in general was a nice challenge for everybody. This is what we are here for. So if there is no other question, I think I would like to thank you all again, Masimba in particular. Oh, thanks. Oh, this is from the supplementary data. Okay. And yes, so Roman, I don't know if we have a date for the next journal club. Roman is not there. Okay. Roman, tu nous entends? Oui, j'imagine. Roman? Okay. Tu m'entends, Roman? Okay. I don't know what's happening with the audio video today.
Are you hear me?
Oui, Brigitte. Yes, I was asking you if we have another date for the next journal club.
Okay. So next journal club.
Okay, so it's the 11th of March. And well, unless a volunteer manifests, I'm afraid that it's my turn with and I've had the paper in the holster for 2 months on genetic regulation on mitochondrial networks. So I'm afraid it's that
No.
but you can avoid it.
Are you hear me?
Please.
Are you hear me? Hear me?
Yeah.
The next speaker is Owen Lukilik,
You're all saved. Perfect.
the student of Master 2.
Great. So perfect. Okay, we will see the topic later. So thank you very much. Thank you, Masimba. Don't worry about the microphone. These things happen. It was a very interesting topic of discussion, and hopefully it will also spark some ideas on our side on how to improve the understanding of this electrical biomarker. So thanks everybody, and take care. We will see each other in a month. Bye-bye.